Imagine descending into the earth with nothing but a headlamp cutting through absolute darkness. No wind, no birdsong, no distant hum of a city or a road. Just the slow drip of water somewhere far below, and the sound of your own breathing bouncing softly off walls that haven't seen daylight in millions of years. The air is cool and still, carrying the faint mineral scent of ancient stone, and every step forward feels like a step backward in time. This is where the story begins. Somewhere deep inside a cave system, scientists were doing what scientists do in places like this. Cataloguing, sampling, mapping the invisible geometry of an underground world. It is patient, careful work, the kind that rarely makes headlines. Most of what they find confirms what they already suspected. A familiar fungus, a known species of bat. The slow geological grammar of stalactites doing what stalactites have always done. But then, something stopped them. In a pool of water so still it looked like polished glass, or perhaps tucked into a crevice in the rock, or clinging to a surface where almost nothing should be able to survive. They found something that did not fit. A creature. Alive. Present. And yet by every rule of biology and geography and evolutionary history that scientists had carefully assembled over centuries, it seemed as though it simply should not be there. Not in that cave. Not at that depth. Not in that part of the world. The discovery raised a question that is quietly one of the most compelling kinds of questions science can ask. Not how does something work, but how did it get here, and what does its presence tell us about a world we thought we already understood? That question is what we're going to spend some time with tonight. This is a story about caves and the strange worlds they conceal. It is a story about a species that appears, on the surface, to be an impossibility. A living thing found so far outside its expected range, so deep inside the earth, that its very existence forces researchers to rethink some of their assumptions about life, about time, and about the hidden connections that run beneath the landscapes we think we know. It is also, like all the best historical mysteries, a story that is still unfolding. Scientists are still piecing together the how and the why, and that uncertainty, that open edge where knowledge meets the unknown, is part of what makes it worth sitting with slowly in the quiet before sleep. So if you're tucked into bed right now, or drifting somewhere comfortable, or maybe listening with your eyes already closed, welcome. You're in exactly the right place for this kind of story, the kind that gives your mind something gentle to turn over, something curious to rest on, without needing to reach any urgent conclusions tonight. Before we go any deeper, a small and entirely optional invitation. If you feel like it, leave a comment letting us know where in the world you're listening from and what time it is there. There is something quietly wonderful about knowing that people are settling in for the night in so many different corners of the earth, all drawn to the same strange story from the dark. Whether it's midnight in Melbourne, or early evening in Edinburgh, or a sleepless 3 a.m. somewhere in between, we're glad you're here. Now, let's go a little deeper, because to understand why this particular discovery matters, we first need to understand something about caves themselves, and the remarkable rule-bending worlds they quietly contain. Most of us, if we think about caves at all, picture them as empty places, dark, cold, hollow, geological accidents essentially. The kind of space that exists only because rock happened to wear away in a particular pattern over a very long time. We imagine them as absences rather than presences, gaps in the earth, places defined by what they lack. And it's true that caves lack quite a lot. They lack sunlight, which is not a minor thing. Sunlight is the engine beneath almost every food chain on the surface of the earth. Plants capture it and convert it. Animals eat the plants. Other animals eat those animals. The whole intricate cascade of life that we tend to think of as simply normal. The forests, the meadows, the ocean surface shimmering with phytoplankton, is ultimately powered by that one relationship between living things and light. Remove it entirely, and from a surface perspective, life should more or less stop. Caves also tend to have remarkably stable temperatures. Not warm, usually. In temperate regions, the deep interior of a cave system often hovers somewhere around the average annual surface temperature of the surrounding area, which can mean a cool, unvarying stillness, regardless of whether it is summer or winter above. That stability sounds almost pleasant until you consider what it means for any creature trying to live there. Temperature variation is actually a signal. It tells organisms when to breed, when to hibernate, when food is likely to be available. Strip away those seasonal cues, and time itself becomes strangely formless. And then there is the matter of nutrients. In a world without sunlight to anchor a food chain, the available energy is sparse and unpredictable. What does exist often enters the cave from outside, carried in by water, blown in by air currents, brought in by the animals that move between worlds, like bats returning from their nightly hunts above ground. The rest is microbial, chemical, slow, chemical. There are organisms in deep cave systems that derive energy from the minerals in the rock itself, a process called chemosynthesis, which sounds like something from a science fiction story but is simply what life does when it finds an environment without obvious options. It invents another way. This is the first thing worth understanding about caves. They are not dead places. They are places where life has had to get extraordinarily creative. The isolation is what makes that creativity possible and necessary. When a population of any organism becomes cut off from the rest of its species, evolution has something to work with. Genetic drift begins to accumulate changes that would otherwise be smoothed out by interbreeding with a larger population. Natural selection presses hard on whatever traits help survival in this specific environment and relaxes entirely on traits that no longer serve any purpose. Eyes, for instance. Many cave-adapted species have reduced or entirely absent eyes, not because darkness damaged them, but because maintaining functional eyes costs biological resources, and in the dark those resources are better spent elsewhere. Over enough generations, eyes become an expense a cave creature can no longer afford. Now here is where the timescales start to feel genuinely difficult to hold in the mind. These processes do not happen quickly. They happen across thousands of generations, tens of thousands of years, sometimes far longer. A species that became isolated in a cave system during the last ice age, which ended roughly 11 or 12,000 years ago, has had enough time to change in ways that might make it almost unrecognizable compared to its surface relatives. And some cave systems have been isolated for much longer than that, far longer. This is why scientists sometimes describe caves as time capsules. The geological formations inside, the stalactites and stalagmites, the layered flowstones, the crystalline deposits of minerals, record environmental history in extraordinary detail, changes in rainfall, shifts in climate, the movements of ancient water tables, all of it encoded in the slow architecture of a rock. And alongside that geological record, the biological one. Species that were once widespread across a landscape might have retreated underground as climates shifted and surface conditions changed, surviving in cave systems long after their surface counterparts vanished entirely. Finding one of these species is, in a sense, like finding a letter written in a very old handwriting, evidence of a world that no longer exists above ground, preserved by the quiet stability of the dark. From a surface perspective, survival strategies in caves can look almost like magic. Creatures that live for decades on almost nothing. Metabolism slowed to a pace that seems closer to minerals than to animals. Species that have shared every feature we associate with typical life. Colour, vision, the rhythms of day and night, and replaced them with something almost alien. Long, delicate sensory structures, extraordinary sensitivity to water movement or chemical traces, the ability to locate a single food source in absolute darkness with a precision that seems improbable, until you remember that precision, in that environment, is the difference between survival and extinction. What caves are, really, is a reminder that the rules of life as we observe them on the surface are not universal rules. They are local conditions. They apply here in this brightness, in this abundance, in this particular relationship between sunlight and soil and season. Change the conditions radically enough, and life responds, not by giving up, but by becoming something genuinely new, something that could only have existed in that specific darkness, over that specific stretch of time, shaped by pressures that most living things will never encounter. And it is in exactly that kind of place, improbable, ancient, hidden, that our particular discovery was waiting. Before we can understand what was living in this cave, it helps to understand what the cave itself has been doing for the past several million years. Because caves do not simply exist. They accumulate. They record. They hold on to things with a patience and precision that no human archive has ever quite matched. Think about what happens when water moves through limestone. It carries dissolved carbon dioxide, which makes it slightly acidic, and that acidity very slowly dissolves the rock it passes through. Over thousands of years, what begins as a hairline crack becomes a passage. A passage becomes a chamber. A chamber begins to fill with the secondary formations, the stalactites hanging from the ceiling, the stalagmites rising from the floor, that most people associate with caves. These formations are called speleothems, and they are not decorative. They are documents. Each layer of a speleothem was deposited by water at a specific moment in time, and the chemistry of that layer reflects the conditions above ground at that moment. A period of heavy rainfall leaves a different chemical signature than a drought. A shift in temperature changes the isotopic ratios of the minerals being deposited. Scientists can core into a stalagmite much the way they core into an ice sheet or a tree trunk, and what they find is a stratified record of past climates stretching back hundreds of thousands of years. Uranium-thorium dating, a radiometric technique that compares the decay of uranium to its daughter products, can establish ages with remarkable precision, sometimes within a few hundred years of a date that is 500,000 years old. That precision matters. It means the cave is not just telling a story, it is telling a story with timestamps. But speleothems are only one layer of the archive. Below them, mixed into the sediments that have accumulated on the cave floor, is another kind of record entirely. Sediments and caves can preserve pollen grains, which tell scientists what plants were growing above ground during any given period. They can preserve the bones of animals that fell through sinkholes, were carried in by predators, or simply lived and died underground. In some caves, the sediment record spans tens of thousands of years of local ecosystem history. Extinctions captured in real time, shifts in fauna corresponding almost exactly to the climate changes recorded in the speleothems above. It is possible to read the same moment in history from two completely different angles in the same chamber, simply by looking up and then looking down. Then there are the signs of the cave's own geological life. Ancient flood lines, watermarks left high on walls when the water table rose far above its current level, tell of periods when the entire system may have been submerged. Breakdown piles, where sections of ceiling have collapsed, record seismic events, or the slow surrender of rock to gravity over geological time. Some caves show evidence of ancient rivers that once ran through them and have since vanished, their channels smoothed and abandoned, the water redirected somewhere else by shifts in the landscape above. A cave that appears still and silent has often had an extraordinarily violent past, and the walls are covered in evidence of it for anyone who knows how to read the language. All of this is relevant to the question of how a particular organism could come to exist in a cave and stay there long enough to become something genuinely new. Because caves are not static environments, and the conditions that allow a species to persist underground can appear, disappear, and reappear across very long timescales. A cave that is cool and perpetually humid today may have been warmer and drier at a different point in history, or flooded to the ceiling, or connected to other cave systems by underground rivers that no longer flow. The biography of a cave and the biography of its inhabitants are not separate stories. They are the same story told from different perspectives. This is also where uncertainty becomes important to sit with honestly. The cave record is extraordinarily detailed in some respects and frustratingly silent in others. Speleothems can tell you a great deal about rainfall and temperature. They cannot tell you what a species was doing while those conditions were changing, or whether a particular lineage survived a particular drought by retreating deeper into the system, or how many times a population may have come close to extinction before stabilizing again. Sediment records are similarly partial. Preservation depends on chemistry, on moisture, on the specific conditions of each individual deposit. What survives in the fossil record of a cave is not a complete account. It is whatever happened to fossilize, in whatever sediment happened to remain undisturbed, in whatever section of the cave happened to be excavated. The gaps are real, and the scientists working with this material are generally careful to say so. What the cave record can suggest, and this is significant, is which parts of a landscape retained relatively stable conditions during periods of major climatic upheaval. Certain cave systems appear to have maintained temperature and humidity within narrow ranges even through ice ages, even through the dramatic shifts of the Pleistocene, when ecosystems that had existed for millions of years were reorganized almost beyond recognition. For a species already adapted to underground life, or in the process of becoming so that stability would have been enormously significant. The surface world was changing faster than many lineages could adapt. The cave was not. This is the context into which the discovery we're building toward must be placed. Not just a dark hole where something unusual turned up, but a geological entity with a long and complex history. A history that created, over an immense span of time, exactly the kind of isolated, stable, resource-limited environment where evolution can produce things that seem, at first encounter, to have no right to exist. The cave was ready long before the scientists arrived. It had been keeping its record quietly, for longer than our species has existed to keep its own. When the first description finally appears in the scientific literature, it does not begin with wonder. It begins with directions, a set of coordinates, the elevation above sea level, the length and orientation of the entrance passage, the average temperature and humidity measured over several visits, the kind of details that tell another researcher, decades later, how to find the same place and recognize it as the same environment. Only after that scaffolding is in place does the narrative narrow, moving from the scale of the whole cave to a particular corner of it. The organism that would eventually be called a new species was not discovered in a moment of revelation. It was noticed almost in passing, during a broader survey of the cave's invertebrate fauna. The team working there had come primarily to sample sediments, to collect microfossils and water chemistry data. Biological specimens were on the list, but they were not the headline. The cave, remember, was already famous in certain circles for its paleoclimate record. The speleothems had been dated and redated. The stalagmites were celebrities. The living things were, at first, supporting characters. Still, cave biology has its own rules, and anyone who spends time underground learns to scan for motion in the beam of a headlamp. On one of those routine sweeps, along a low, damp passage at the fringe of a larger chamber, a researcher saw something that did not immediately fit into the mental catalogue of expected cave life. It was small, pale and still at first glance. Many cave organisms are, but there was something about its outline that made the eye stop. Limbs that seemed just a little too long, a body that seemed just a little too slender or too broad for the group it vaguely resembled. It was resting on a thin film of moisture that coated a ledge of rock, occupying a sort of micro-terrace where water from a ceiling drip spread, pooled, and then trickled down into a narrow crevice. In the notebook that day, the description is almost curt. Unusual form observed on wet ledge at 230 meters from entrance. Side passage B. Collected 3 specimens. Morphology atypical for known troglobitic group in region. The brackets here stand in for a taxonomic identity that will be teased apart later in the paper. At that first encounter, the exact classification is less important than the fact that it did not match the expectation for that particular stretch of cave. The initial observations focus on morphology, because that is the language taxonomists are trained to think in. How many segments? How many legs? The shape of the antennae, if there are antennae. The arrangement of spines or bristles. In many cave-adapted animals, eyes are reduced or absent, pigment is lost, and limbs are elongated. All of those traits can be present in different combinations, and the specific pattern can suggest relationships to surface-dwelling relatives. What stood out here was not just the typical hallmarks of underground life, but the way they were combined. For its group, the eyes were more regressed than usual, the extremities more attenuated, the body proportions shifted. There were structures where none should be, or the absence of structures where all the described relatives had them. Even before the specimens were preserved, the field notes mention marked depigmentation, complete lack of ocular pigment, an exaggerated elongation of appendages relative to trunk length. Behaviour was noted too in the unhurried observational style that good fieldwork encourages. Rather than fleeing rapidly when disturbed by light or vibration, the organisms moved with a slow, almost deliberate motion, probing the wet surface with fine sensory structures. They seemed reluctant to leave the narrow band where the film of water was thickest, avoiding both the drier rock above and the flowing trickle below. One specimen paused for several minutes with the front half of its body slightly raised, as if sampling the air currents invisible to human senses. This tiny ledge, in other words, was its own little world inside the larger cave. Not a random stretch of wall, but a habitat pocket defined by specific conditions, a constant drip, a persistent sheen of water thin enough to oxygenate but not so deep as to wash small bodies away, organic particles periodically delivered from higher passages and trapped in the sticky film. The cave as a whole might have been stable, but within it certain surfaces and corners were more stable than others, more predictable in the resources they offered. That is where the organism was found. What made it especially puzzling was the broader biogeographic picture. The group to which it tentatively belonged was well known from surface habitats many kilometers away, at different elevations and in different climatic zones. Related cave forms had been described, but from other karst systems with clearer connections to modern rivers or ancient valley networks. On a map of known distributions, this cave was a blank space between clusters of records. There were no documented populations of its supposed relatives in the immediate surroundings, above ground or below. An experienced biologist learns to be suspicious of blank spaces on maps. Sometimes they mean a species truly is absent. More often, they mean no one has looked carefully. Even so, the ecological mismatch was difficult to ignore. The chemistry of the water film, the microclimate of that passage, the apparent diet inferred from the fine structure of the mouthparts seen under a hand lens, all of it suggested a lineage that had been specialized for this kind of environment for a very long time. This did not feel like a recent arrival that had stumbled into the cave and begun to adapt. It felt entrenched. And yet in the first formal report the language is cautious to the point of understatement. The authors do not announce a discovery that overturns textbooks. They write of a morphologically distinctive population, of forms not readily assignable to any described species, of a taxon that, if confirmed as novel, would represent a significant extension of the known range of its group. The words "if confirmed" do a great deal of work. This is how science talks when it is standing at the edge of something it does not yet fully understand. Anomaly rather than revolution, unusual rather than impossible. The report notes the organism's apparent isolation, its extreme adaptations, and the absence of close analogues in nearby sites. But each observation is wrapped in conditional phrasing, given context, and immediately followed by alternative interpretations. Perhaps, they suggest, these specimens represent a highly modified population of a known species that has colonized the cave relatively recently, evolving cave-associated traits in a small number of generations. Perhaps they are part of a cryptic species complex already present on the surface, but overlooked because the differences are subtle outside the cave environment. Perhaps, less likely but not excluded, the individuals were transported into the system from another, as yet unsurveyed subterranean network via ancient or seasonal groundwater connections. From the beginning, multiple hypotheses are laid out like paths in a dark chamber, none yet illuminated all the way to the end. The organism might be an evolutionary relict, a survivor from a climate regime recorded in the stalagmites above its ledge. It might be a case of convergent evolution, where similar pressures in different caves have sculpted similar forms from unrelated ancestors. It might even be, at least in principle, an introduced species carried in by human activity, though the authors are careful to mark this as unlikely, given its morphology and restricted microhabitat. This deliberate proliferation of possibilities is not hedging for the sake of politeness. It is a reflection of how incomplete the first glimpse always is. A handful of specimens from a single corner of a single cave cannot, by themselves, reveal the true age of a lineage or the full outline of its distribution. What they can do is signal that something is out of place with respect to current maps and current stories, and that the maps and stories may need to be revised. In the pages that follow, the authors begin to assemble the toolkit for that revision. Careful measurements, comparisons with museum specimens, genetic analyses where material allows. But before any of that can happen, the first report has to do something more modest and more radical at the same time. It has to say, in carefully measured prose, here is a living thing in this exact spot at this exact time that does not fit where we thought the boundaries were. To say that something does not fit is only the beginning. The next question is whether it fails to fit because it is truly new, or because the existing categories are misdrawn, or because the observers do not yet know the full diversity that has already been described. Turning an oddity on a wet ledge into a recognized species is less a single leap, and more a long, methodical climb. It starts with the least glamorous part of science, the notebook and the label. Those first few specimens still damp from the cave air are not just dropped into a jar and forgotten. Each one is given a context that will follow it for the rest of its strange afterlife. A code is written on waterproof paper. The cave name, the date, the passage, the exact distance from the entrance, the drip rate if it was measured, the temperature, the humidity, the time of day. Often there are notes about what else was nearby. Other invertebrates, fungal films, algae, bat guano, fragments of leaves. The more detailed the field notes, the richer the story that can later be reconstructed. Back at the surface, the specimens move from the world of a rock and darkness into the world of vials and cabinets. Preservation comes first. Depending on the organism and the era, that might mean ethanol of a particular strength, a fixative to stabilize delicate tissues, or a careful drying and mounting on slides. One part of the sample may be set aside to remain intact as a physical voucher, another may be reserved for dissection. A third portion, if the collectors were thinking ahead, is preserved specifically for molecular analysis, in a way that protects the DNA from immediate decay. This splitting of a tiny body into multiple futures is deliberate. A specimen is more than an object, it is raw material for different kinds of questions. To be useful over decades, it has to be prepared in ways that allow those questions to be asked later, sometimes with tools that do not yet exist at the time of collection. Once preserved, the organism begins to be translated into numbers and words. Under a dissecting microscope, the overall shape is described in far greater detail than the hurried notes underground could manage. Lengths are measured with micrometers. Ratios are calculated. Limb length to body length, the width of a particular segment relative to the one in front of it, the angle at which a spine projects from the cuticle. For groups where male genital structures or the arrangement of sensory hairs are diagnostic, those parts are dissected out, cleared, and mounted so that their outlines can be seen clearly. This is the craft of morphology, and it has its own vocabulary, honed over centuries of looking at small, complicated things. A single sentence in a taxonomic description can encode a surprisingly rich pattern. Which edges are serrated, which surfaces are smooth, how many subdividing branches a hair has before it ends, whether an eye is truly absent or just reduced. All of this is compared, trait by trait, with the published descriptions of known species. In the past, that might have meant leafing through printed monographs and keyed identification guides, following a branching path of choices. Does the animal have 3 segments here or 4? Are the terminal appendages forked or simple? And so on, until a name is reached. Today, it also means searching digital databases, looking at high-resolution images from other collections and cross-referencing geographic ranges and ecological notes. Very often, what first appears unusual in the field turns out, under this closer scrutiny, to fall within the variation of a known species. Cave populations, in particular, can exaggerate certain traits that are only weakly developed in surface relatives, making them seem more divergent than they really are. The job of the taxonomist is to be as strict with themselves as possible, to resist the temptation to name something new when a more prosaic explanation will do. Because of that, the next step is almost always comparison with physical reference material. Descriptions on paper can be ambiguous, specimens in drawers are more concrete. This is where museums and research collections become central. In climate-controlled rooms in universities and natural history institutions, cabinets hold the types, the individual specimens on which existing species names are based. A researcher who suspects that the cave organism might represent a new taxon will arrange to visit these collections, or to borrow relevant material on loan. They place the unknown next to the known, rotate them under the same light, examine the same structures in the same way. If the published key says that species A has a particular arrangement of bristles on the third leg, the taxonomist checks the actual holotype of species A to confirm that this is truly so, and not an artifact of an older microscope or a descriptive oversight. Sometimes, those visits result in the realization that a supposedly new form has already been named, perhaps in a little cited paper from a century ago. Other times they have the opposite effect. The differences become starker when the comparison is direct. Alongside this traditional body-based work, a parallel line of evidence has become increasingly important. DNA. In essence, sequencing involves extracting the genetic material from a bit of tissue, a leg, a piece of muscle, a cluster of cells, and reading out the order of the chemical letters that encode its biology. No single gene can fully define a species, but certain regions of the genome change at rates that make them useful for distinguishing lineages and for inferring how those lineages are related. For many animal groups, researchers often target standard markers, a segment of mitochondrial DNA perhaps, or a particular nuclear gene. The sequence obtained from the cave organism is then compared to sequences in global databases. If there are close matches, that suggests a relationship to already characterized species. If the sequence falls on its own isolated branch in the genetic tree, that supports the idea that it represents a distinct lineage, not just a local variant. Importantly, DNA data do not replace morphology, they complement it. A surprising genetic result can prompt a re-examination of subtle physical features that were overlooked. Conversely, a striking morphological novelty might be revealed through sequencing to be the product of rapid adaptation in a lineage that is otherwise nested within a familiar group. The back and forth between what can be seen and what can be read from molecules is where much of the modern work in identification happens. All of this, however, depends on trust. Trust that the specimen sequenced is what the label says it is, and that the sequence belongs to that organism alone. To build that trust, contamination and misidentification have to be guarded against at every step. In the cave, tools may be rinsed between samples, and collectors avoid scooping up multiple species into the same vial unless they are thoroughly sorted later. In the lab, DNA extractions are often done in clean spaces, physically separated from where amplified genetic material is handled, to reduce the chance that an amplified fragment from one project will drift into another. Negative controls, tubes that go through all the same steps without any actual tissue, are processed alongside the real samples. If they show a sequence, it is a warning that stray DNA is present somewhere in the pipeline. On the morphological side, misidentification is checked by having more than one pair of eyes look at the material. A specialist in the group might be consulted, independently of the original team. Measurements are repeated. When a feature is described as unique, it is cross-checked against as many specimens of related species as possible, not just against the published words about them. The institutional structures of science are designed in part to formalize these habits of skepticism. Once the researchers believe they have a case, that the cave organism combines a distinctive body plan with a distinctive genetic profile, and that it does not match any named species in the literature or the collections they have consulted, they write a manuscript. In it, they lay out the evidence step by step, where the specimens came from, how they were preserved, what they look like, how they compare with close relatives, how their DNA sequences cluster on a phylogenetic tree. That manuscript is then sent to a journal, where it is read not by an anonymous public but by a small group of peers, often chosen because they have spent much of their own careers working on similar organisms. These reviewers look for gaps. Did the authors overlook an obscure reference? Did they fail to compare with a particular museum collection known to hold relevant material? Are the measurements based on enough individuals to account for natural variation? Are the genetic analyses robust to different methods of constructing the tree? Sometimes the response is that more work is needed. Additional specimens from other parts of the cave, checks against collections in another country, sequencing of extra genes. Sometimes the reviewers point out that the supposed new species falls within the variation of an existing one once juvenile forms or sexual dimorphism are considered. And sometimes, after revisions and clarifications, the conclusion that this is indeed a new taxon withstands the scrutiny. Even then, the process does not culminate in a dramatic declaration so much as in a formal description and a name, printed in small type in a specialized journal. A holotype specimen is deposited in a museum, its label bearing the same coordinates and date first scribbled in the cave. Paratypes, additional individuals that show the range of variation, may be placed in other collections so that future workers across the world can check their own finds against them. Identification, in this sense, is not a single moment of discovery but a chain of careful acts. Observing, recording, preserving, comparing, doubting, confirming. Each link is mundane on its own. Together they allow that pale, slow-moving creature on a wet ledge to move from the realm of anecdote into the shared, cumulative architecture of knowledge, where others can find it again, test new ideas against it, and perhaps be puzzled anew by the way it fits, or does not fit, into the larger patterns of life without light. To understand why the new species seemed so out of place, the researchers had to step back from its bones and genes and ask a simpler, harsher question. How does anything live here at all? On the surface, the answer is so familiar that it almost disappears from notice. Green plants catch sunlight, turn it into sugars, and everything else, from grazing deer to fungi to humans, builds its own life on top of that captured energy. Break the chain down far enough and you find photosynthesis at the base, quietly underwriting the entire visible world. In the cave, that contract is broken. Past a certain point from the entrance, there is no dawn and no dusk, no dimmer switch of twilight, only a steady blackness. No green leaves can grow in that blackness, no moss, no algae film on a rock, no pale shoots groping toward a window. If the ordinary rules applied strictly, if every ecosystem were required to start with a sunlit leaf, then the deep cave would be a dead zone, interrupted only by gravity and dripping water. It is not dead. It is simply on a different kind of budget. Instead of drawing a regular paycheck from the sun, the cave runs on irregular deliveries and careful thrift. Ecologists sometimes describe these places as detritus-based systems, built not on fresh living photosynthesis, but on leftovers imported from somewhere else. One of the most important delivery services is the wind and the weather above. During storms, water rushes into sinkholes, along swallow holes in the bedrock, down narrow fissures. As it goes, it carries things with it. Fragments of leaves, twigs, pollen grains, a drowned insect, a bit of soil. Over time, these scraps accumulate in quiet pools and on ledges where the current slackens, forming thin, dark carpets. To a hungry springtail or beetle larva, this is not debris, it is a pantry. Bats, too, play the role of unknowing benefactors. Many caves shelter large colonies, sometimes thousands or tens of thousands of animals, that pour out at dusk to feed in the forests and fields above. They return heavy with the insects they have eaten, and what they cannot digest falls to the floor as guano. A thick guano pile is a concentrated rain of energy from the surface, rich in nitrogen and phosphorus. Entire miniature communities form within it, fungi and bacteria, mites, beetles, fly larvae, all feeding in one way or another on what the bats have left behind. Other animals contribute in smaller but still crucial ways. A salamander that spends part of its life outside the cave might bring in a meal and drop the uneaten portion. Floods may sweep in the carcass of a mouse or a bird, or the broken shell of a snail. A cricket that strays too far from the entrance and dies becomes a sudden rare feast for scavengers that may go weeks without such bounty. Even the bare rock is not always as barren as it looks. In places where water seeps constantly across walls and ceilings, fine microbial films develop, slippery biofilms which to the human touch are just slime, but in fact are dense communities of bacteria and other microorganisms. They can grow by metabolizing dissolved organic material in the water, remnants of everything that has washed in from soil and plant roots above. grazers scrape at these films with specialized mouthparts, turning a surface that seems sterile into a grazing ground. All of these are different faces of the same pattern, energy that was first captured by sunlight somewhere else, arriving in the dark as cast-offs and waste. The cave is, in that sense, an economy of imports, and the currency is carbon trickling downward. There are, however, a few places underground where life is not tied so directly to distant leaves and grass blades. In some caves, particularly those connected to deep groundwater or hydrothermal systems, chemicals bubbling up from the bedrock itself can become the fuel. Here, instead of photosynthesis, chemosynthesis takes center stage. Certain bacteria can take energy from oxidizing compounds like hydrogen sulfide, methane, or reduced iron, and use that energy to build organic molecules from inorganic carbon. They are in effect playing the same role as plants, but their sunlight is chemical disequilibrium, the contrast between what is dissolved in one bit of water and what is dissolved in another. In a few well-studied caves, thick mats of such bacteria form on submerged rocks, or hang from ceilings as pale streamers. Grazers and filter feeders have evolved to specialize on these mats, and an entire food web unfolds from a process that never sees the sky. These chemosynthetic oases are striking, but they are also patchy and rare, tied to particular geological conditions. Most caves, including the one where the strange species was found, depend primarily on the more prosaic influxes of detritus from above. Whatever the source, energy in caves is almost always scarce and unpredictable. A flood might bring a sudden surge of material and then nothing at all for months. A bat colony may collapse or move to another roost, and the guano that sustained generations of invertebrates dwindles to a few scattered pellets. Unlike a meadow in summer, there is no guarantee of steady production. Under those conditions, living things tend to take on a different pace. Many obligate cave species grow slowly. With limited food, cells divide less often, bodies add tissue cautiously, and reaching adulthood can take years instead of months. Some cave crustaceans and insects have been estimated, through mark and recapture studies and growth ring analyses, to live far longer than their close relatives on the surface. Lifespans stretch, not because time itself changes underground, but because it is safer in an uncertain economy to invest in maintenance and survival than in rapid turnover. Reproduction follows the same logic. With little energy to spare, many cave organisms produce fewer offspring at a time, often larger or better provisioned eggs rather than vast clouds of tiny ones. Brooding behaviours, where parents guard or carry their young, are more common in these dark spaces than in some comparable surface habitats. It is as if each new individual is a precious, expensive project that cannot be risked lightly. Metabolisms are tuned to frugality. Some cave animals can endure long periods without eating at all, slowing their activity, reducing movement to a minimum, waiting until the next insect carcass or pulse of leaf litter appears. Muscles are thin, pigment is discarded, eyes are reduced or lost, not only because they are unnecessary in the dark, but because building and maintaining them costs energy that cannot be justified. When researchers first sketched the energy flows in the cave where the impossible species was collected, they were effectively trying to write its budget. How much organic matter normally enters this system in a year? How many bats roost here, and how thick is the guano layer beneath them? How large are the seasonal floods? How much leaf litter do they carry? How quickly is it decomposed? How many layers of consumers sit on top of that base, each taking its small share? Within that accounting, the new organism looked extravagant. Its size, its apparent abundance on certain ledges, and its life history traits, inferred from dissections and observations, all seemed out of proportion to the trickle of energy available. On paper, there did not seem to be enough carbon falling from above, or enough chemical fuel welling up from below to support such a population over long spans of time. This is where the language of a species that should not exist takes on a specific technical meaning. It is not that the animal violates any known physical law. It is that, under the most straightforward assumptions about what comes into the cave and how it is used, the numbers do not add up neatly. Something in the ledger is missing. Perhaps the cave receives more energy than the first surveys captured. An unnoticed seasonal flood route, a hidden guano mound in an upper chamber, a steady drizzle of fine organic particles from a crack high in the ceiling. Perhaps the species' lifestyle is stranger than assumed, tapping into a food source that had been overlooked, or living at a pace so slow that its true demands are far lower than a surface analogue would suggest. Whatever the ultimate resolution, the initial sense of impossibility was a clue, not a wall. It pointed the investigators away from microscopes and gene trees, back into the cave itself, to follow the hidden paths of water and rock and time that might be quietly feeding life in the dark. If the question of energy is about the cave's income, then the next question is about its residents' expenses. How do you make a life on a budget so tight that, by surface standards, it barely qualifies as an income at all? One answer is written directly on the bodies of the creatures that have committed to the dark. Biologists have a word for animals that spend their entire lives in caves, never venturing out into daylight. Troglobites, from the Greek for cave dweller. Over time, many of these obligate cave species develop a suite of traits so distinctive that they almost deserve their own category of creature. Collectively, those traits are called troglomorphic, cave-formed, as if the rock itself had a hand in sculpting them. The most striking of these, and the one that often catches human visitors off guard, is the loss of eyes. In the entrance zone of a cave, where daylight still reaches in shifting beams, you might meet animals that have normal vision or only slightly reduced eyes. Deeper in what cavers call the dark zone, eye after eye disappears. Some species keep tiny vestigial eye spots buried under skin, the last trace of structures their ancestors once used. Others lose them entirely. In a few, the skull bones grow smooth and unbroken where eye sockets would be, as if the head had been redesigned from the blueprint up. At first glance, this can look like a story of deprivation, of something essential being taken away. But in evolutionary terms, it is also a story of release. Eyes are expensive. They are complicated organs that must be built during development, wired into the nervous system, protected from damage, supplied constantly with oxygen and nutrients, and connected to a brain region capable of interpreting their signals. In a place where photons never arrive, all of that cost brings no return. Across generations, mutations that reduce eye development or alter the way energy is allocated to the growing embryo are no longer punished. They can persist, accumulate, and eventually turn a functioning eye into a ghost of its former self. Around the same time, pigment fades. Many deep cave animals are pale, milky, or translucent. Their internal organs faintly visible through thin skin or exoskeleton. The reds, browns, and greens of their surface relatives drain away. Pigment, too, is an investment. It protects against ultraviolet radiation, provides camouflage, plays roles in temperature regulation and signaling. Underground, where there is no sunburn and no visual signaling, much of that becomes irrelevant. The genes that once coordinated pigment production can drift, their careful tuning loosened. Cells that would have devoted resources to melanin or other colour molecules can pour them into something else, or simply not spend them at all. But if eyes and pigments shrink, other senses swell. Run a fingertip gently along the body of a blind cave shrimp or insect, and you may feel a forest of fine hairs and bristles, each more than an ornament. These are mechanosensory organs, finely tuned to vibrations and currents. In fish and amphibians, the lateral line, tiny canals and pits studded with sensory cells, can become more elaborate. Antennae lengthen, joints become more flexible, and in some crustaceans, entire segments seem devoted to reaching into the water, sampling it for hints of movement and chemistry. Chemosensation, the ability to detect dissolved substances in water or airborne molecules in air, becomes particularly important. In the dark, smelling and tasting are not luxuries. They are the primary way to map a world. Many cave beetles, millipedes, and crustaceans have proportionally larger chemosensory organs than their closest surface kin. Under the microscope, the tips of their antennae or mouthparts bloom with sensilla, tiny sensory hairs connected to nerve cells that respond to specific chemicals. In some species, these structures are so numerous and densely packed that the organ looks fuzzy, like a miniature brush. Taken together, these changes make troglobites look uncanny, even alien, to eyes accustomed to birds and deer and city pigeons. Blind, pale, elongate, with oversized feelers or limbs, seen side by side they can seem like variations on a single strange theme. Yet when scientists examine their genealogies, they often find that these lookalikes are not close relatives at all. A blind white fish in a Balkan cave may be more closely related in genetic terms to a normal-eyed riverfish upstream than it is to a blind white fish in Mexico. A pigmentless beetle in a Spanish limestone system may share more ancestors with sun-loving beetles in nearby forests than with another cave beetle on the other side of the mountain range. This is where another key concept enters the story: convergent evolution. Convergence is what happens when unrelated lineages, facing similar challenges, independently stumble on similar solutions. Wings in bats and birds, streamlined bodies in dolphins and ichthyosaurs, succulent stems in cacti and some old-world euphorbias. Each case involves different branches of the tree of life bending toward the same function. Caves are convergence machines. Remove light, restrict food, stabilize temperature and humidity, and the rules of survival shift in consistent ways, no matter what the starting organism looked like. Time and again, in different continents, in different rock types, from ancestors as varied as insects, crustaceans, fish, and salamanders, evolution has pared away eyes and colour, stretched bodies and antennae, and amplified the senses that still bring useful information in the dark. From the outside, the repetition can feel almost eerie, as if the cave itself imposes a uniform shape on life. From the inside, for the creatures themselves, it is simply what works. One of the most important and least visible of these adjustments lies not in limbs or senses, but in the invisible chemistry of metabolism. On the surface, in rich seasons, many animals live by the motto of grow fast, reproduce fast. Their metabolisms are set to move quickly, fueling bursts of activity, rapid development, frequent reproduction. If an individual fails, others can take its place. The system can afford waste. In energy-poor caves, that strategy is dangerous. A body primed to burn fuel at a high rate may starve between unpredictable meals. Instead, many troglobites show signs of metabolic downshifting. Laboratory measurements, where cave animals are gently placed in respirometry chambers and their oxygen consumption is tracked, often reveal lower basal metabolic rates than in their surface relatives. These are not animals idling at the red line. They are idling low, engines barely humming, so that they can run for a very long time on very little. Some can go astonishing stretches without feeding. Cave fish in controlled studies have survived months without food, losing weight, slowing their movements, but remaining alive and responsive. Certain cave arthropods show similar endurance, entering states of torpor where motion nearly ceases until a new food resource appears. Muscles may be less bulky, digestive tracts longer and more efficient, tissues biased toward maintenance rather than rapid turnover. At the same time, heightened chemosensory abilities help make the most of any opportunity. Detecting a faint trace of amino acids drifting downstream from a decaying insect, or the subtle change in odour that signals bat guano in the next chamber, can mean the difference between another week of hunger and a rare, rich meal. Noses, antennae, and taste receptors are, in effect, the cave equivalent of a well-developed financial sense, attuned to the tiniest fluctuations in a sparse market. Navigation, too, runs on different cues. Where we would reach for a flashlight and a compass, a cave animal threads its way through a maze of rock using a combination of vibration, airflow, and chemistry. A sudden tremor in the water, carried along a gallery, might signal the movement of a potential predator or mate. Tiny changes in current, felt along whisker-like appendages, trace the outline of unseen boulders and ledges. Air itself carries information. In passages with multiple branches, slightly cooler or moister air may drift from one direction, warmer or drier from another, hinting at a larger chamber or a connection to the surface. Some bats and insects are known to use airflow as a guide. It is plausible that many small troglobites do the same, even if the details of their perception are still being teased apart. Chemical trails are another layer on this invisible map. Many invertebrates on the surface use pheromones and scent marks to guide social behavior, finding mates or food sources. In the compressed geography of a cave, where populations can be small and scattered, such signaling may be even more crucial. A faint trail left by a wandering individual could help another of its species locate it in the dark, or lead scavengers to a rare carcass before microbial decay erases the opportunity. Put all these pieces together: reduced metabolism, heightened chemosensation, reliance on vibration and airflow, the sacrifice of eyes and pigment, and the animals that seemed, at first, to be violations of the rules start to look like meticulous accountants of energy and information. They are not exempt from constraints. They are specialists in living inside them. When scientists described the new impossible species from the cave, part of what unsettled them was that it seemed to have gone even further down this path than expected, or in a direction that did not match the standard cave-adapted template. Its proportions, its sensory structures, its inferred lifespan or reproductive strategy did not fit neatly on the graphs drawn from other troglobites. It occupied, in anatomical and ecological space, a point that looked empty before. The phrase "should not exist," repeated in headlines and summaries, can easily sound like an accusation aimed at the animal itself, as if it were breaking some universal law. In practice, among the researchers who use it most carefully, it is often a confession. What they mean, more often than not, is, this form of life lies outside the expectations we built from limited data. It violates a model, not a law of physics. It shows that the constraints we thought we understood, the minimum energy needed to support such a body, the range of shapes that convergent evolution tends to favour, the sensory trade-offs that seem inevitable in the dark, were drawn too tightly. Every troglomorphic creature in its own way is a quiet argument against assuming that the familiar is the only possible. The cave system that produced this particularly puzzling species was not defying biology. It was exploring a corner of biological possibility that had not been seriously mapped. To understand how that exploration unfolded and why this lineage ended up with such an unlikely combination of traits, the investigators had to look beyond the present moment. Bodies carry history in their proportions, in their genes, in the way they develop from egg to adult. So do the rocks around them. The next step was to ask what kind of world first gave rise to this strange specialist of the dark, and how that world has changed since then. When biologists first begin to suspect that a cave animal is older, in evolutionary terms, than the surroundings it lives in today, a particular idea tends to surface in their conversations. It has a slightly romantic name: refugium. A refugium is, at its core, a shelter. Not a shelter from predators or from storms, but from time periods. The surface of the Earth is not a steady place. Over tens of thousands of years climates swing. Ice advances and retreats. Forests swell and then thin to scattered copses. Lakes expand into shallow inland seas, then contract into salty basins. Deserts bloom and wither. For the species living through these shifts, survival is often less about outrunning change, and more about finding pockets where change arrives late, or only partially, or not at all. Caves, for all their darkness and strangeness, are very good at being such pockets. Deep underground, temperatures hover close to the local annual average, fluctuating only slightly across the year. Humidity is high and steady. Wind is muted. Sunlight, and everything tethered to its daily and seasonal rhythms, stops at the threshold. To us this can feel like deprivation, like the stripping away of all the cues by which we tell time. To an organism faced with a world above that is frosting over, or drying out, or burning in seasonal extremes, it can feel like stability. This is the essence of the refugium hypothesis as it applies to caves. During times when the surface becomes harsh or hostile for a given species, some individuals retreat or are stranded in underground spaces that blunt those extremes. There, buffered from the worst, they persist as the climate outside rolls through its cycles. To see why this matters, it helps to zoom out, very far, and imagine the last few million years not as a flat line, but as a waveform. During the Quaternary period, a span that includes the last 2.5 million years or so, the Earth's climate has been marked by repeated glacial cycles. Driven by subtle changes in the planet's orbit and tilt, amplified by feedbacks in ice, oceans and atmosphere, great sheets of ice have sprawled out from the poles and then retreated again, over and over. In the colder phases, glaciers advanced over northern continents, sea levels dropped, and belts of climate shifted toward the equator. Where temperate forests had stood, steppe or tundra might take hold. Regions that were once mild enough for broadleaf trees might find themselves locked in permafrost. In the warmer interglacial periods, like the one we live in now, ice retreated, sea levels rose, and forests recolonized ground they had lost. Rivers cut new courses, lakes reappeared in ancient basins, animals and plants expanded their ranges, often in directions that look, on a map, like the reverse of the earlier retreat. Layered on top of these glacial pulses were other, more local shifts, centuries-long droughts, intervals of increased monsoons, changes in prevailing winds, volcanic winters, and regional warming trends. Over geological time, coastlines migrated, mountain ranges continued to rise or erode, and land bridges appeared and vanished. For a lineage trying to maintain itself through all this movement, the map of what counts as good habitat was constantly being redrawn. Sometimes species tracked these movements. Forest animals followed trees as they crept up valleys or down slopes. Fish and amphibians rode connected river networks as meltwater carved new paths. Seeds blew, rafts of vegetation drifted, birds flew. But not every population could move with the ideal pace or direction. Barriers cropped up, a newly arid corridor blocking moisture-loving plants, a glacier plugging a valley, a newly formed canyon slicing across an ancestral route. When that happened, some organisms died out locally. Others, caught in corners where conditions stayed just tolerable enough, clung on. In karst landscapes, regions where soluble rocks like limestone and dolomite are riddled with caves, sinkholes and underground streams, those corners often lay beneath the surface. Imagine in one glacial period a humid temperate forest reaching down into what is now a subtropical zone. Streams run clear through shaded ravines, carrying cool, oxygen-rich water. Along those streams lives a small, unassuming creature, perhaps a crustacean or a salamander that can use both surface and underground creeks, slipping into cracks and fissures when drought or predators press too hard. As the climate cools further, the forest fragments. Trees thin and retreat upslope. The streams shrink, their summer flows reduced. Patches of habitat that once formed a continuous band now resemble islands in a widening sea of unsuitable ground. The individuals closest to cave entrances, feeding in twilight pools or breeding in sheltered springs, find that those subterranean spaces change less than the sunlit reaches of their range. The water remains cool, the humidity high. Food may be scarcer, but the basic physical environment does not swing wildly from one decade to the next. Over thousands of years, as one glacial episode piles on another, these underground pockets become the main place where that lineage persists. On the surface, its relatives disappear, squeezed out by cold, by drought, or by competing species better tuned to the altered conditions. Underground, in the buffered climate of the cave system, A remnant population carries on, slowly adapting to the very shelter that saved it. This is one way a relic species can form, as the last surviving thread of a once more widespread fabric. A relict in this sense is not just any old species. It is one whose present distribution and ecology make the most sense when you consider past environments very different from today's. It may have close relatives in the fossil record but no living cousin in neighbouring rivers or forests. Its nearest modern kin might live hundreds or thousands of kilometres away, in a climate that seems surprisingly mismatched to its current surroundings. Caves are known to harbour such relics. In parts of the southeastern United States, for instance, blind cave crayfish belong to lineages whose surface-dwelling relatives are either scattered and distant or absent from the surrounding waters. In the Dinaric karst of the Western Balkans, the famous olm, Proteus anguinus, a pale, blind salamander, dwells exclusively in subterranean streams, even though its closest relatives are surface-living amphibians now found elsewhere. Its body carries echoes of a time when cool, shaded waters were much more extensive at the surface in that region. In Europe's cave beetles, genetic studies have identified lineages that appear to have been isolated underground since at least the last glacial maximum, roughly 20,000 years ago, and in some cases across multiple glacial cycles. Their distribution maps look like ghost outlines of former forests or ancient river systems, preserved in the present as a scattering of dots in separate cave systems. These relics matter because they act as biological core samples. Where a lakebed preserves layers of sediment and pollen, a relic species preserves, in its genes and anatomy, a record of where and under what conditions its ancestors once thrived. By comparing relics with their living and fossil relatives, scientists can reconstruct past landscapes, which valleys were wetter, which plateaus were forested, which river basins were connected. In the case of the impossible cave species that unsettled its discoverers, one line of thinking went like this. Perhaps this animal is not in fact a rule-breaking novelty born entirely within the cave system as we see it today. Perhaps it is a survivor from an older surface world, one whose climate would make its peculiar combination of traits seem far less strange. If, for example, its physiology suggests comfort with cooler, more stable water temperatures than nearby surface streams now offer, or if its reproductive cycle seems tuned to rhythms that no longer exist above ground, that might fit a refugium story. If its closest living relatives inhabit regions that were once connected by wetter corridors, a chain of springs, a vanished river, a now buried karst conduit, that is another hint. Genetic data can sharpen or blur this picture. If DNA comparisons show that the cave species diverged from its nearest relatives hundreds of thousands or millions of years ago, long enough to have lived through several glacial cycles, that strengthens the case for an ancient isolation. If, instead, the split appears very recent, more in line with a few tens of thousands of years or less, the refugium hypothesis becomes more cautious. Perhaps the cave is a short-term shelter in an ongoing cycle, rather than a vault from a distant era. Fossil evidence can weigh in as well, finding remains of similar animals in old surface deposits, lake sediments, river terraces, ancient spring mounds, near the cave suggests that the lineage once had a broader, daylight-facing presence. The absence of such fossils does not disprove the idea, but their presence is a powerful corroboration. On the other hand, if the only representatives of this form, living or fossil, are strictly subterranean, scattered across caves that were never directly connected by surface waters, then a different scenario might be more likely. A lineage that originated underground, dispersing through hidden routes rather than retreating from a disappearing surface. Even the cave itself offers clues. Speleothems, stalagmites, stalactites, flowstones carry chemical signatures of past rainfall and vegetation above. By sampling them, scientists can infer when the region was wetter or drier, warmer or cooler. If the appearance of the puzzling species in the cave's sediments coincides with a known period of surface stress, a prolonged drought, a sharp cold snap, that temporal overlap can be suggestive. None of this by itself can turn the refugium hypothesis from a story into a certainty. Evolution rarely leaves tidy labels on its pathways. Instead, researchers compile strands of evidence, genetic divergence times, anatomical comparisons, fossil occurrences, cave climate records, and see whether, woven together, they resemble the pattern of a long-term shelter from vanished conditions, or something else. For a sleep-bound listener, the image that may linger is simple. Somewhere in the deep, a creature goes about its hushed life, its every movement a living residue of rains that no longer fall, forests that no longer shade these valleys, rivers that no longer run in the open air. It is less an intruder from another world than a quiet holdout from an earlier version of this one, and if caves can serve as time capsules for species, preserving lineages that once swam in open rivers or skittered through leaf litter, then the stone around them may have done something similar for water itself, locking away flows that no map now shows. To follow the history of this strange animal further, scientists had to look not only at the refuge it found, but at the hidden roots that might still connect its dark domain to the vanished landscapes of its past. Karst is a quiet word for a restless kind of landscape. On the surface, karst regions can look almost ordinary. Rolling hills, scattered trees, perhaps a dry valley or two that seems curiously empty of running water. But underfoot, the rock is behaving in a different way from the granite of old mountains or the sandstones of broad plateaus. It is dissolving. Limestone, dolomite, and related rocks form from ancient seas, from the compressed shells and skeletons of marine life, from layers of carbonate mud laid down over millions of years. They may look solid now, but rainwater, made slightly acidic by carbon dioxide in the air and in the soil, seeps into tiny fractures and begins to melt its way through. Drop by drop, century by century, those fractures widen into hairline cracks, then fingers, then tunnels. Streams vanish into the ground where their channels intersect these growing voids. Whole river systems slip underground, reappearing kilometers away as powerful springs. The result is a three-dimensional maze of shafts, conduits, and chambers, threaded through the bedrock, often stretching far beyond the small patch of ground where you can see an actual cave entrance. To walk across a karst plateau is, in a sense, to stroll over the roofs of rivers. Because the water is doing the carving, those underground networks rarely respect the boundaries humans draw on maps. A surface ridge that looks like a divide between two river basins may, below, be pierced by a tunnel that quietly steals water from one side and carries it to the other. A valley that appears dry may, in wet seasons, become a roaring temporary river that disappears into a sinkhole at its lower end, feeding a spring in an entirely different watershed. For organisms that use water as their road, these twisted, unseen networks can matter more than the visible landscape. In such systems, the line between surface stream and subterranean river is often just a short fall through a crack. During floods, when rains are heavy or snowmelt surges, the distinction blurs even further. Imagine a storm sweeping over a karst plateau. Thin soils saturate quickly. Water pours into swallow holes, into the grates of caves, into every fissure that will take it. Surface creeks swell, then overtop their banks carrying leaves, silt, insects, fish, and anything else caught in their flow. Where the stream bed intersects a sinkhole or a swallow point, the water spins down like liquid rope, dragging its suspended cargo with it. Eggs laid on submerged stones, larvae drifting in the current, tiny crustaceans grazing in the upper layers, all can be swept underground in such pulses. Some will die in turbulence or darkness. Others will be flushed back to the surface through spring outlets downstream. But a few may find themselves deposited in quieter backwaters, a side pool in a cave stream, a sump at the base of a shaft, a slow eddy in an underground river where the current slackens. During dry periods, the direction of movement can reverse. Springs that normally gush outward may fall silent, while water tucked in deeper conduits continues to flow, now drawing on reserves stored in distant, unseen chambers. In these moments, certain underground stretches can become more hospitable than the fragile surface trickles they once drained. For organisms that already tolerate low light or intermittent food, this can turn a one-way tumble into the dark into the first step of a new home. Hydrologists and biospeleologists talk about subterranean dispersal corridors to capture this idea. Instead of thinking of caves as isolated pockets, little dots on a map, each sealed away from the others, they picture chains of voids linked by water pathways. A corridor in this sense is not a single grand tunnel you could walk from end to end. It is a distributed route made of porous joints, small conduits, sumps, and intermittent streams that, taken together over time, allow material, and sometimes living things, to move from one region to another. Sinkholes act as gateways into these corridors. Some are dramatic pits, obvious even to a casual passerby. Others are modest depressions in a field, only truly active when heavy rains transform them into temporary ponds. To the eye, they are mere landscape curiosities. To water, they are doors. In flood events, those doors swing wide. Surface and subsurface become parts of the same churning system, and any organism whose life stage coincides with such a pulse has a non-zero chance of being admitted and carried along. This is one way that species can end up in places that seem inexplicable if you only trace their likely paths above ground. A salamander found in a cave well beyond the current edge of its surface range might be the descendant of many such flood-driven accidents, with only a handful of pioneers surviving and reproducing in some hidden chamber where the conditions happen to suit them. From the perspective of the scientists studying our impossible cave creature, the karst beneath its valley offered a similar invitation to think in 3 dimensions. On the surface, the animal's known relatives lived in streams several valleys away, separated by dry ridges and basins that showed no sign of connecting waterways. By the usual logic of rivers and watersheds, those populations should have been isolated from one another for a very long time. Yet the genetics suggested a more recent separation than the visible landscape would allow. To probe that contradiction, researchers did what cave hydrologists have done for more than a century. They tried to follow the water, even where they couldn't see it. One classic tool is dye tracing. In its simplest form, a harmless fluorescent dye is poured into a sinkhole, a losing stream, or a cave river at one point in the system. Downstream, at suspected springs, wells, or cave passages, scientists place charcoal packets or sensors that can detect even faint traces of that colour. Over days or weeks, as the dye moves through the rock, its eventual appearance tells you that 2 points you might have thought separate are, in fact, connected by a hidden path. When this method is repeated at different times of the year, under wet and dry conditions, a more nuanced picture emerges. Some routes may only activate in flood, carrying pulses of water, and whatever lives in it, through conduits that are otherwise dry. Others flow steadily but slowly, suggesting long underground journeys that can take weeks. Paired with dye work are careful measurements of water chemistry and temperature. Each catchment area imparts a slightly different signature to its water. Ratios of dissolved ions, trace elements, even the stable isotopes of oxygen and hydrogen. If a spring that appears in one valley carries the unmistakable chemical fingerprint of rainfall from a different plateau, something unseen is ferrying that water beneath the hills. In the region of our cave species, such studies have gradually redrawn the hydrological map. Springs that local people long assumed were fed by the nearest surface streams turned out, under dye tests, to be receiving water from distant basins. Small sinkholes in dry pastures were quietly connected to roaring outlets far away. Rivers that seemed to terminate at a cliff base were actually sending part of their flow through a network of conduits that arced under an entire mountain range. When biologists overlapped these subterranean watersheds with the locations of related species, patterns began to emerge. Populations that looked isolated on a topographic map now sat along the same hidden conveyor belts. Caves that hosted similar assemblages of animals, despite being in different surface drainages, lined up along inferred underground routes. In such a context, the misplaced cave animal no longer needed a miraculous origin story. It could have arrived the same way leaves and silt and twigs did, as cargo in an underground river, carried over distances no surface map would encourage you to imagine. Perhaps during some particularly wet interval in the past, a sinkhole in the ancestral range of its surface. Relatives swallowed not just water and debris, but a clutch of eggs or a brood of juveniles. Swept down into darkness, most perished. A few, deposited in a low-current alcove of the receiving cave system, survived long enough to feed, to mature, to breed. Over time, cut off from regular exchange with their source population, by shifting flows, and by the difficulty of making that journey in reverse, they became established. From there, Other subterranean corridors might have stepped them along, cave to cave, much as island chains can ferry terrestrial animals across oceans over evolutionary timescales. The dispersal would be rare, stochastic, often failing, but given long enough, even a low probability route can leave a mark in the form of a puzzled scientist holding a specimen that should not be there. This way of thinking does not exclude refugia, it extends them. A cave can be both a shelter from changing climates and a junction in a hidden transport network, a station on an underground railway that occasionally carries life from one climatic province to another. To understand how extraordinary the cave species' current home really is, and how it might have been reached, the researchers had to accept that their initial map, the simple overlay of rivers and ridges, was, in an important sense, incomplete. The true outline of the animal's world lay not just on the surface, but in the dark, water-filled veins of the karst beneath. Only after that realization could they begin to see how often our diagrams of isolation and connection, of islands and mainlands, of centres and peripheries, are drawn on the wrong plane. When biologists first plotted the locations of our cave animal and its supposed relatives, the pattern looked simple enough. Clusters here, a gap there, a lonely dot far away. A tidy distribution, the kind that fits easily into a paper or a textbook diagram. But those dots came from places where someone had actually gone, knelt in cold water, and looked. Most caves on Earth have never been visited by a scientist. Many have never been mapped, never even been given a name beyond a local nickname or a note in a shepherd's memory. Some are known only as bad air in a hollow, a chill draft from a crack, a place where animals vanish. In regions riddled with karst, the underground voids vastly outnumber the entrances we notice, and even among the entrances we notice, only a fraction are ever explored in a systematic way. A cave that floods seasonally may be inaccessible at the very times when its biology is most active. A sump might block further passage, hiding kilometres of habitat beyond a single pool of opaque water. When your knowledge rests on such partial forays, the range of a species can easily look far smaller and more broken than it really is. The absence of records along a stretch of valley may reflect nothing more significant than the absence of cavers with notebooks. In the early years of subterranean biology, this kind of undersampling was simply accepted as an unavoidable limitation. You collected where you could, in the caves that were known and relatively safe. The resulting maps felt authoritative, their labelled dots suggesting neat zones of presence and absence. From those patterns, people drew conclusions about isolation, speciation, and dispersal. Only later, when more caves were explored, did awkward complications appear. A supposedly endemic species turning up hundreds of kilometres away, or a gap in the distribution filling in as soon as someone finally crawled through a tight passage into a neighbouring system. For our cave species, something like this happened on a smaller scale. At first it was known from a single chamber in a single cave. Then a second population appeared in a nearby system that had been visited many times before without anyone noticing. Only when a researcher lingered, sampling methodically through seasons rather than in a single trip, did the small, pale animals reveal themselves. This raised a quiet question. How many other caves, already known, held life that had simply been overlooked? Part of the problem lies in the animals themselves. Subterranean species are often small, translucent, and shy of disturbance. They may be active only at certain times of day, or only during particular flow conditions. Some cling under stones or hide in thin films of water that a casual glance slides right past. Others look, at first, indistinguishable from more common, surface-dwelling relatives. Such familiarity can be deceptive. For decades, taxonomists sorted cave fauna largely by morphology, the shapes of limbs, the structure of jaws, the arrangement of bristles, the proportions of body segments. These are powerful tools, but in the underground world, evolution often pushes different lineages towards similar solutions. Eyes shrink or vanish, pigment fades, appendages lengthen, bodies become more slender, more flexible. The result is convergence, unrelated species growing to resemble one another in gross outline. Where convergence hasn't fully smoothed away the differences, the remaining distinctions can be subtle. A slightly different curve of a claw, a minute extra tooth, a pattern in reproductive structures invisible without careful dissection. Under pressure to catalogue diversity with limited time and resources, it was easy to group such creatures under a single familiar name. In this way, what seemed like one widespread species might actually be a mosaic of cryptic species, lineages that are genetically distinct, with their own histories and ecological nuances, but that hide behind a shared appearance. The impossible distribution of our cave creature began, in part, as a problem with these names. Animals from distant caves were placed into the same taxonomic box because, to the eye and under a hand lens, they differed little. Only when genetic sequencing became part of the toolkit did deeper divisions emerge. Mitochondrial DNA, nuclear markers, and later more comprehensive genomic scans revealed lineages that had been evolving separately for hundreds of thousands, sometimes millions of years. What had been thought of as one adaptable, far-ranging species resolved into a cluster of related, more localized species, each tracing its own path through the geological and climatic shifts of the region. In the case of our cave animal, those data did something paradoxical. They made its presence in the wrong cave both more puzzling and easier to explain. Puzzling because its closest relatives turned out not to be the surface populations that had first been assumed, but another hidden population itself long misassigned. Easier to explain because that pairing aligned better with what the underground hydrology was now telling researchers about past connections. Peeling back the layers of cryptic diversity also exposed another issue: where and how people had chosen to look in the first place. Exploration and record-keeping are never neutral. Early cave biology in many regions unfolded in places that were already popular with tourists, or close to universities, or accessible to organized caving clubs. These tended to be near towns, along established roads, or in landscapes that governments considered deserving of detailed mapping. Remote karst plateaus, politically marginal borderlands, territories of indigenous peoples, or areas labelled uninteresting or dangerous, were often left off the scientific itinerary. Local knowledge of caves, sometimes rich, sometimes detailed over generations, might be ignored or minimised if it did not come packaged in the forms recognised by official surveys. In archives, the bias continued. Specimens from famous caves were carefully labelled, preserved, written about. Finds from obscure holes in the ground might be mentioned in passing, without precise coordinates, or left in drawers with only vague locality notes. When later researchers tried to reconstruct distributions, they leaned heavily on the better documented sites. Gaps in the record looked like gaps in nature. These historical choices shaped the earliest maps of subterranean life, and so shaped the question scientists asked. If most known occurrences of a group clustered in one region, that region was called the centre of diversity, the likely cradle. Outlying dots became problems to solve, anomalies requiring special explanation, even when they simply reflected where someone had managed to go. As technology changed, the edges of this picture began to soften. Genetic tools did more than reveal cryptic species. They also allowed for the identification of organisms from trace amounts of material. A sliver of tissue from a long-preserved specimen, a few cells caught in a water sample, even fragments of DNA shed into the environment, could carry information about organisms that were never directly seen. Environmental DNA, eDNA, grew out of this insight. Instead of relying solely on catching or observing animals, researchers filter water, soil, or sediment, and then sequence whatever genetic material is present. Specific sequences can be matched to known species, while unmatched sequences hint at lineages not yet described. In cave streams, where water flows continuously through habitats that are difficult, dangerous, or impossible for humans to reach, eDNA acts like a passing whisper from unseen chambers. A sample taken at a spring may contain genetic traces of species living far upstream in the dark. What appears on the filter can suggest that somewhere, beyond a sump or behind a too-tight fissure, a population exists that no one has ever collected. For our cave species, eDNA surveys in the region's karst springs revealed its signature in places where no physical specimens had yet been found. That did not prove thriving populations in each of those spots, but it did undercut the earlier impression of a single, isolated stronghold. The animal's world was wider than the handful of caves that humans had entered. At the same time, advances in imaging and mapping transformed how caves themselves were understood. Laser scanners, ground-penetrating radar, refined photogrammetry, and improved diving equipment allowed cave explorers to chart conduits once written off as inaccessible. Three-dimensional models stitched together from multiple expeditions showed how apparently separate systems might hook into each other just beyond a sump or a choke. Biologists could overlay genetic data onto these new maps, watching clusters of related lineages track the shapes of now revealed tunnels and chambers. Features that had looked like hard boundaries on old maps, blank spaces between known caves, often turned out to be crossed by networks of voids. Taken together, these developments forced a reconsideration of words like impossible. A salamander found far from its supposed range, a blind fish in a basin where no such species had ever been recorded, a crustacean that seemed to have leapt a mountain chain. Each of these became less a violation of natural order and more a signal that the map, in one way or another, was incomplete. Sometimes the incompleteness was physical: undiscovered caves, unmapped conduits, unseen sinkholes and springs. Sometimes it was biological: cryptic species tangled under a single name, traits that misled the eye. Sometimes it was historical: records clustered where people had chosen to look and to write, empty where they had not. In the story of the cave species that should not exist, the growing realization was not that nature had broken its own rules, but that humans had been working from a partial ledger. Our diagrams of ranges and barriers, of ancient refugees and dispersal routes, have always been, in the end, hypotheses drawn from what we happen to know. Caves, with their hidden spaces and selective accessibility, expose this more starkly than many environments. When a find seems to contradict everything the map says, one response is to search for heroic narratives, improbable journeys, freak events, mysterious catastrophes. Another, quieter response is to turn back to the map itself and ask where its lines come from, what they leave out, and how our tools and habits of looking have shaped them. In that sense, every impossible creature in the dark is not just a biological mystery, but a reminder that the boundaries we draw around the living world are often more fragile and more provisional than the rock above our heads. To understand how our cave animal came to be where it is, and to be what it is, you have to slow your sense of time down until it barely seems to move at all. On the surface, we're used to thinking in quick cycles. A storm builds and breaks in an afternoon. A forest can be logged in a year. A town can double in size between one census and the next. Even when we talk about evolution, the stories we tell are often about rapid change. Finches switching beak shapes over a few decades, insects evolving resistance to a pesticide within a human lifetime. Caves ask for a different rhythm. Once a conduit has formed, once water has carved a path and the ceiling has stopped collapsing in a catastrophic way, the environment inside can become remarkably stable. Temperature swings narrow to just a few degrees across the whole year. Light never appears, so it never has a chance to vary. Storms mute into distant sounds. Seasons show themselves only as slight changes in drip rates or nutrient trickles. In a place like that, evolution does not freeze. It stretches. Mutations still occur in the DNA of our cave species. Tiny copying errors as cells divide, as eggs and sperm form, as generations replace one another. But those generations may turn over more slowly than in their surface relatives. Food is scarce, so growth is slow. Lifespans can be long. Populations are small, tucked into isolated pools and side passages, never very numerous even at their peaks. Small populations do something strange to the way evolution feels. When there are only a few hundred or a few thousand individuals in a population, chance events carry more weight. A mutation that appears in one animal's germline might vanish instantly if that animal happens, by bad luck, to leave no surviving offspring. Another mutation, which offers no particular advantage or disadvantage, might spread simply because the individual carrying it leaves descendants by good fortune alone. This is genetic drift, the slow, random wandering of gene frequencies over time. In large populations, selection tends to overpower drift for changes that matter. In tiny, secluded populations, drift can have a freer hand. Over tens of thousands of generations, it can nudge lineages apart, accumulating quiet differences in the background of their genomes even as their outward forms remain, for a long while, deceptively similar. In caves, that process plays out under the constraint of a high-stability environment. The strong, obvious pressures, no light, limited food, need to navigate in tight spaces, push species in certain directions. Eyes shrink, pigment fades, antennae stretch. Those changes may happen relatively early in a lineage's underground history, because natural selection is clearly acting on them. After that, the pace of visible change slows. A population that has already abandoned vision and melanin has fewer big, obvious steps left to take. It still evolves, but much of that evolution hides in invisible places, in metabolic tweaks, in subtle shifts of development, in changes to reproductive timing, and in neutral stretches of DNA where mutations drift without immediate consequence. For scientists trying to reconstruct the history of a cave species, this creates a paradox. The animal may look like it has always been there, frozen in some ancient shape, yet its genome carries a record of countless tiny steps. To read that record, researchers turn to something called a molecular clock. The idea, in essence, is simple. If mutations accumulate in DNA at a roughly steady rate over long spans of time, then the number of differences between two lineages can be used as a kind of stopwatch. Count the changes, estimate how fast the ticks have been, and you can infer how long ago those lineages diverged from a common ancestor. In practice, it is never quite so clean. Mutation rates are not carved in stone. They can vary between groups, between genes, even between parts of the same genome. Some stretches of DNA are under strong functional constraint. They change very little over millions of years. Others are more permissive. Their sequences can shuffle rapidly without harming the organism. So researchers calibrate. They take lineages whose divergence times are constrained by fossils or by known geological events, and they use those anchor points to estimate how many molecular ticks correspond to 1 million years in that group. They test different genes, compare models, and attach uncertainties to every estimate. When they apply these tools to our cave animal and its closest relatives, the numbers that come back carry wide bands of possibility, plus or minus hundreds of thousands of years, sometimes more. But even with those uncertainties, patterns emerge. The deepest splits between major lineages often fall in periods when the landscape above was undergoing grand rearrangements. Mountain belts rising, rivers shifting course, sea levels dropping and rising again. Shallower splits cluster around later events, changes in glacial cycles, the capture of one river by another, the uplift of a plateau that subtly redirected underground flow. This is where the timescales of biology and geology begin to interlock. Imagine for a moment the region above our caves not as a static map, but as a film played in very slow motion. A river, once flowing toward one sea, gradually incisors its bed, carving deeper and deeper into the rock. On the other side of a ridge, another river does the same, chewing backward through its own drainage basin. Over millions of years, one cuts just far enough to intercept the headwaters of the other. A single stream is captured, and its waters begin to flow along a new route. On the surface, the channel might wander into a different valley. Underground, that rerouting can be more dramatic. Water that once seeped into a particular limestone layer and fed a given cave system may be redirected, starving some passages of flow and invigorating others. Old conduits can dry out, new ones can be etched, connections can be severed without any human eye ever seeing the process. For a subterranean animal that depends on those waters, such a change can be equivalent to the sudden appearance of a mountain range. A once continuous population may find itself split into two basins, two cave systems that no longer share water. Over time, the lineages on either side begin to drift apart genetically, their molecular clocks ticking away in parallel but no longer synchronized by gene flow. When scientists today compare their DNA and run their models, they may find that the divergence time between those lineages aligns within its margin of error with the estimated date of that river capture event. The biological clock and the geological clock point, tentatively, to the same moment. Something similar happens with uplift. As a mountain range rises, or as a plateau is gently hoisted upward by deep forces, the gradient of rivers changes. Streams cut deeper, springs migrate, recharge zones for aquifers shift upslope. Caves that were once at the water table may become perched, hanging dry above new, lower conduits. Others may flood permanently. Again, each of these transitions redraws the underground map from the perspective of an animal living there. A population that once had a continuous habitat along a valley may find parts of that realm stranded, transformed into isolated pockets. Over long spans of time, those pockets can become cradles for distinct lineages, each following its own quiet trajectory. When genetic data suggests that two cave populations parted ways around the same time, that a section of the landscape was uplifted or tilted, researchers see a possible connection. The rock moved, the water followed. The animals, in their own slow fashion, responded. None of this is precise in the way a watch is precise. Molecular clocks are blunt instruments for measuring deep time, especially in groups that lack a rich fossil record. Error bars are broad. Different genes can tell slightly different stories. It's entirely possible that some divergences we line up with particular geological events are in reality offset by hundreds of thousands of years. But even with that fuzziness, the broad picture is striking. Our cave species, and its hidden cousins, did not simply appear in their present refuges by accident last week, or last century, or even in the span of human history. Their lineages reach back into climates that felt entirely different on the surface. Times when glaciers advanced and retreated, when forests spread where grasslands now are, when coastlines sat tens of kilometers away from their modern positions. The caves themselves, or at least the voids they occupy, may predate those climatic swings. Water has been threading its way through the same belts of limestone for millions of years, adjusting its exact routes as the land flexed and rivers found new paths. In that restless frame, the apparent stillness of a cave stream is an illusion of our own short attention. For a viewer lying in the dark, listening, it can be soothing to let the mind drift along these extended timescales, to imagine mutations arriving one by one in a small pale population, each one a tiny note in a melody that only becomes audible over immense spans. To picture rivers slowly erasing and redrawing their basins, carving opportunities and barriers that no human eye will ever directly witness. In that perspective, the impossible distribution of our cave animal becomes less a puzzle about how something got somewhere, and more a meditation on how long it has been quietly adapting to changes that, to us, would be imperceptibly slow. Deep time in this story is not a violent sequence of cataclysms. It is a series of almost undetectable shifts. Water levels dropping by centimeters, rock dissolving grain by grain, a gene frequency inching its way from 1% to 2. Over millions of years, those increments add up to whole mountain chains, whole new cave systems, whole new species. By the time we arrive with our headlamps and sample bottles, the major acts have already unfolded. We catch the lineage in a single frame, at a particular moment, in a particular pool. Behind it lies a history that stretches beyond any human calendar, written not in ink on paper, but in the shapes of tunnels and the sequences of base pairs. Ahead of it, if conditions allow, lies more of the same. Slow, quiet change, ticking away in the dark, on timescales that feel almost unreal compared to the restless tempo of our own lives. From here, it becomes easier to see why the presence of a human in that underworld is such a recent, fragile thing in comparison, and why, when we do leave our own marks in caves, they stand out so sharply against the backdrop of geological and evolutionary calm. From the cave's perspective, human beings arrive like a sudden flickering weather event. For millions of years, its tunnels have known only the slow pulse of water and the faint movements of resident life. Then, in a span so short it barely registers on a geological scale, a new creature begins to appear at the entrances carrying something the cave has never seen before, its own portable sun. A headlamp beam seems harmless. So does a flashlight, or the glow of a phone screen raised to take a photo. Yet for a place that has spent its entire existence in perfect darkness, even a momentary burst of light is a shock. Bats rouse from torpor when beams sweep across their roosts. Algae, which would never have been able to grow in the absence of light, can suddenly find a foothold on damp walls near tourist paths. In some heavily visited caves, green films and tiny ferns cluster around lamps, forming what guides sometimes call lampenflora, little oases of photosynthetic life that, from a cave ecosystem's point of view, are as alien as plastic. Light is only the most visible piece of our footprint. Each person who steps underground carries a cloud of invisible companions and traces: skin flakes, hair, fibres from clothing, dust caught in shoe treads, microbes from their own bodies, cosmetics, and household detergents that cling faintly to the skin. Collectively, this detritus is sometimes called lint, but that word suggests something soft and trivial. In a confined, stable environment, it becomes an input, an unexpected trickle of nutrients and organisms that the cave did not evolve to handle. On a popular route, the floor can be dusted with fine, pale film that is essentially human fallout. Bacteria and fungi that would never make it into that particular chamber on their own are introduced sometimes repeatedly, sometimes in huge numbers during busy seasons. They settle onto speleothems and into pools, competing with or preying upon native microbes that have spent millennia finely tuned to the cave's chemistry. Even our breath changes things. Every exhalation carries carbon dioxide and water vapour, in the open air that simply folds back into the vast atmosphere. In a tight passage or a small chamber, especially one with poor ventilation, CO2 levels can rise noticeably when groups linger. Over time, repeated visits can nudge the average composition of the cave air upward. Carbon dioxide dissolves in water to form a weak acid. In caves, this chemistry is central to how limestone is dissolved and formations are built. When the balance shifts, even slightly, the rates of dissolution and precipitation can change. Stalactites that have grown in one set of conditions for thousands of years may find themselves in a subtly different chemical bath. In extreme cases, delicate calcite needles can dull, cloud over, or stop growing altogether. Body heat adds a further twist. Cave temperature is famously stable, usually hovering near the average annual temperature of the surface above. That stability is part of what makes the environment so distinctive. A handful of people in a large cavern barely disturb it, but in narrow, enclosed spaces or in caves designed for mass tourism, the cumulative warmth from lights, electrical equipment, and hundreds of human bodies can raise the local temperature by a degree or more. From our perspective, 1 degree seems negligible. From the viewpoint of a cave-adapted organism, that shift can be enormous. Remember how precisely tuned many subterranean species are. They have evolved in an environment where weather barely exists, where 10,000-year climate trends feel like the only noticeable changes. A slight increase in temperature can alter oxygen solubility in water, speed up metabolism, change microbial community structure, and influence how much organic matter decays before anything can eat it. Our cave animal, with its slow life cycle and small population, has no easy way to accommodate rapid shifts. It cannot move to a new valley or climb to a cooler hillside. Its entire world is this specific flow, this chamber, this seep. If the chemistry or temperature of that patch changes beyond its narrow tolerance, there is nowhere else for it to go. This is why, in the last few decades, cave scientists have begun to think more carefully about what it means simply to walk into these spaces with a notebook and a vial. Fieldwork at first glance looks inherently disruptive. You enter the cave with bright lights, you tromp across a stream bed, you scrape a sample of mud from the wall, you collect a few animals for study. Each of those actions taken once is a tiny event. Taken many times over, or repeated by many people, they can add up to real damage. Modern ethical practice tries to shrink each footprint as much as possible. Before stepping into a new system, researchers typically secure permits from authorities or landowners, and those permits often come with conditions born from hard-earned experience. Limit group size, stick to established paths, avoid sensitive formations, restrict sampling in known habitat of rare species. The goal is not just bureaucracy. It is to ensure that the act of studying a cave does not, in itself, erase the thing that made the cave interesting. Many teams now treat caves like clean rooms in reverse. Instead of protecting themselves from contaminants, they protect the cave. Boots are scrubbed and sometimes disinfected, especially in regions dealing with fungal diseases like white nose syndrome in bats. Gear is decontaminated between sites so that microbes, spores, or tiny invertebrates aren't unknowingly transported from one underground system to another. Sampling strategies have become more conservative. Instead of scooping out entire clusters of organisms, researchers might take only a few individuals, sometimes just tissues or hairs, relying on modern genetic techniques that can work with minuscule amounts of material. Environmental DNA methods, capturing traces of genetic material left in water or sediment, are increasingly used to detect species without ever needing to see or disturb them. There is an ethic at work here that goes beyond simple caution. It recognizes that many cave species live right on the edge of viability. A population of our mysterious animal might consist of only a few hundred individuals in a single stream reach. If a researcher were to collect a large fraction of those for study, or if a sudden spike in visitors were to trample the substrate where they shelter, the impact would not be quickly absorbed because there are no vast reserves elsewhere to recolonize the site. Recovery, if it happens at all, would unfold not over seasons but over centuries, and it is not just scientists and cavers who influence these worlds. Far above, decisions that seem only distantly related to caves ripple downwards. Mining operations that quarry limestone can intersect active conduits, draining or contaminating them. Blasting can alter the hydrology of an aquifer, redirecting flows away from long-established passages. Groundwater extraction for agriculture or urban water supplies can lower water tables, drying subterranean streams that have flowed steadily since long before human settlement. Pollutants travel quickly through karst. Fertilizers, pesticides, industrial effluents, sewage, anything that seeps into sinkholes or percolates through thin soils can be channeled with surprising speed into cave systems. What may appear on the surface as a minor leak or a distant spill can, in the underworld, translate into a sudden toxic pulse through a habitat that has no buffer and no alternate route. Tourism adds yet another layer. Show caves have introduced millions of people to the underground, inspiring awe and often respect. At the same time, unregulated or poorly managed visitation can be rough on fragile environments. Paths widen to accommodate crowds, flatten microhabitats. Handrails gather skin oils and leave dark streaks on formations where visitors touch for luck. Litter accumulates in side passages. Noise and foot traffic can disturb bat colonies, causing them to abandon roosts they have used for centuries. When tourism is carefully managed, with strict limits on visitor numbers, thoughtful lighting, elevated walkways and constant monitoring, it can coexist with conservation and even fund it. When it is not, the cave becomes another exploited landscape, its uniqueness worn down by a steady tide of casual impact. All of this might sound like a catalogue of threats, a list of ways in which humans disturb a realm that functioned just fine without us. There is truth in that. But another way to frame it is as a story about timing. We have arrived very late in the history of these systems. The species we encounter, the blind fish, the ghost white salamander, the strange crustacean that should not exist where it is found, are chapters in a narrative that began long before our species learned to chip stone tools. Their lineages carry memories of past climates and ancient rivers, of mountain belts rising grain by grain. To conserve them is not only to keep a rare organism alive, it is to allow that long story to continue. In practical terms, that means treating caves not as empty playgrounds or convenient drains, but as archives and habitats with their own integrity. It means supporting regulations that limit pollution and unsustainable water use in karst regions. It means asking hard questions about whether a new mine, a new highway, or a new resort will intersect underground systems that we barely understand. For scientists, it means moving through these spaces with a kind of humility that is still relatively new in our history, acknowledging that our curiosity, powerful as it is, does not grant us the right to take without consequence. Each footprint, each sample, each installation of a sensor or a light fixture is a decision about how the future of that cave will differ from its past. For those listening in the dark, drifting towards sleep, it can be comforting to imagine that somewhere below, in hidden chambers, this slow, ancient drama is still unfolding. Our cave animal, and others like it, continue their unhurried lives, feeling only the faintest echoes of the rapid, noisy world above. Whether that remains true is, in some measure, up to us. If we choose to tread lightly, to see caves as more than curiosities or resources, then the underworld can keep writing its story in water and stone and DNA. Our own entry becomes just one chapter, brief, intense, and, with care, not catastrophic. And if we succeed, then future generations, long after we're gone, may still be able to step into these same cool corridors, switch off their lamps for a moment, and feel the presence of a life that has been flowing in darkness since long before any human foot first crossed the threshold. If you trace the path of this story backwards, from a single, fragile creature in a trickle of water, to a notebook in a scientist's hand, to the long corridors of stone that sheltered it, you find yourself standing at a crossroads of explanations. Perhaps this animal is a relic, a survivor from an older climate when rivers flowed differently and shorelines lay in other places, left behind as the world shifted around it. Perhaps it is the product of an improbable journey, carried through flooded tunnels or buried channels that no longer connect on any map, guided not by intention, but by the slow insistence of groundwater. Perhaps, in a quieter way, it tells us that evolution can repeat itself, sculpting similar forms in different caves when conditions converge, so that what looks like a single, impossible lineage might be several, echoing one another in the dark. Each of these ideas has evidence behind it. None of them, on its own, feels complete, so the discovery becomes less like an answer and more like a prism. Shine a question through it, about climate, about movement, about isolation, and the light splits into different colours, each one a partial truth. What makes this single cave animal so unsettling is not just where it lives, or how specialised it is, but what it does to our neat diagrams of the world. Biogeography, the attempt to understand why species live where they live, often looks tidy on paper. Boundaries are drawn, this valley, that mountain range, this river basin. Arrows show how lineages spread or shrink. Regions are named and separated as if life had politely followed the margins of our maps. Then a pale, blind creature appears in a pool where, by those rules, it should never be. Suddenly, old assumptions wobble. Maybe the barrier we thought was absolute was, at some forgotten time, porous. Maybe a cave we'd treated as an isolated pocket is in fact a node in a buried network. Maybe resilience does not always look like abundance or mobility. Sometimes it looks like persistence in a single hidden refuge, holding on through ice ages and droughts while everything visible above comes and goes. In that sense, the animal is both fragile and formidable. Fragile, because its population may be tiny, its tolerance narrow, its home easily disturbed. Formidable, because its very presence implies a lineage that has outlasted glaciers, sea level swings, and continental rearrangements by retreating into stability and staying there, generation after slow generation. It invites us to expand what we mean by survival. Not only the loud, visible kind, where species march across continents, but the quiet kind that takes place in overlooked corners, sinkholes, aquifers, pockets of constant temperature, where life weathers change by stepping out of the main current and waiting. Every time a discovery like this is made, the edge of the known world shifts slightly. A dot appears on a distribution map where there was only blank space before. A genetic tree grows a new branch. A sentence in a textbook changes from never to almost never, from impossible to rare, but documented. History often seems like a story of centres, great cities, famous empires, major events. Yet again and again, it is the margins, the frontiers, the borderlands, the places nobody bothered to survey carefully, that rearrange our understanding when someone finally looks closely. Caves are that kind of margin, turned inward. They remind us that the planet is not fully catalogued, that beneath well-mapped surfaces lie volumes we have barely skimmed. For all our satellites and sensors, our instruments and models, there are still creatures carrying unrecorded histories in their bones and blood tucked away in dark hollows. The broader lesson is a quiet one. Knowledge does not expand like a spotlight sweeping across a stage, illuminating everything at once. It grows along its edges in small increments, wherever curiosity pushes a little farther into uncertainty. A small team squeezes through a tight passage, peers into a dripping chamber, and gently nets an animal no one expected. Months later, in a lab filled with humming equipment and labelled vials, patterns emerge that ask us to redraw bits of our mental world. Not because the animal is famous or dramatic, but because it sits precisely where our explanations begin to fray. There is something soothing in that. It suggests that the unknown is not a yawning void, but a fine mist sitting on the borders of what we already understand, waiting patiently for attention. Each careful observation, each hesitant theory, condenses a drop out of that mist and lets it fall into the slow river of shared knowledge. Curiosity in this view is not a restless demand for spectacle. It is a willingness to linger with puzzles that may never be fully solved, to accept partial answers, to hold several possibilities in mind without forcing a conclusion. Patience turns that curiosity into science. Years of returning to the same cave, counting the same few animals, measuring the same water chemistry, double-checking the same sequences of DNA. Work that is unhurried, repetitive, easy to overlook. Work that over time reveals that a creature that should not exist does exist, and in doing so rearranges our sense of what is possible. As you drift towards sleep, you might imagine that somewhere underground this process continues. Not only the life of the cave animal, moving slowly through its eternal twilight, but also the parallel life of the questions it raises, circulating in the minds of people who will probably never meet it, yet feel its quiet tug on their understanding. The story is not finished. It may never be. And that is all right. Science does not need to close every loop to be meaningful. Its beauty often lies in the open ends, in the way each answer gently points toward new uncertainties, like a passage narrowing into darkness, inviting, but not demanding, that someone one day explore a little farther. If you found this slow journey into the underworld comforting or thought-provoking, you can help these kinds of stories continue. When you like this video, or leave a comment, or subscribe to the channel, you nudge the algorithms just enough to tell them that quiet history and the overlooked lives it contains are worth bringing to more people. If you feel like sharing, you're warmly invited to write where you're listening from, what city, what region, maybe even what kind of landscape lies above you as you listen. Desert, forest, mountains, flatlands, coastline. Somewhere beneath those surfaces, known or unknown, water is moving through stone, and life is finding ways to exist in places we have not yet imagined. And little by little, as patience and curiosity do their slow work, the edges of our map will continue to soften and expand, one hidden chamber, one mysterious species, one quiet discovery at a time.